Thank you very much, Pavel, and good afternoon, everyone. I'm going to be talking about better trials, better answers, designing trials that truly matter. Okay, so this outline my talk, I'm going to walk you through six steps. Pavel told me to talk a bit about my role as a statistician and what I do day to day, as well as some of the things that I've been doing. I thought that I might have to give an introduction to early phase dose finding trials, but I think we've heard quite a lot of that in a non-oncology setting, and maybe I'll focus a bit more on the oncology setting. And then putting novel early phase trial designs into action. So we'll talk a bit about a few case studies, and incorporating patient-reported outcomes in these early phase settings, and then good reporting in protocols, as well as trial results publications, and I'll end off with some key messages. So I'm just going to give you an appetizer of all these six different things, and please feel free to come and speak to me if you're interested to learn more about it. So my role, but first of all, architects designed safe and useful homes. So what does a statistician do? So you can see of a statistician as like building a clinical trial house. They design clinical trials to find reliable answers, whether a new treatment is actually safe and effective. And in this house, there are different rooms. So it could be the trials planning room, the trials operational site, or the data review lab, or the presentation hall. So, and there's a little bits of things that I do day to day. So either I'll be designing a trial, I'll be helping to conduct the trial, like, involving its development of statistical analysis plan, or actually the actual recruitment and tracking recruitment, analyzing and communicating results. So the different things, and you spend different amount of times in different rooms. And I have the pleasure to work with different people and with different disciplines. So I used to work quite a lot, primarily with clinicians and trials team, but increasingly we've been working directly more with patient partners as well. And that's actually been a really kind of like interesting dimension to what I've been doing. And underlying it is a foundation about better methods. So better methods, whether that be in design, conduct, analysis, or communications, we need that foundation. And there's always things that we can do better in each of these different parts of the rooms. Okay, I think most of you will know this very well, so just to run through very quickly, obviously there are 4 different phases in clinical trials. The key objectives are listed above. The ones in orange are normally called the exploratory phase or early phase. Well, then it goes on to the phase 3, which is definitive trial, and then the phase 4, which is looking at long-term efficacy and safety. Now, in the early phase, we've heard quite a bit about it already. So phase 1 is typically about safety and tolerability. And then phase 2 is looking at activity, which could be single arm or which could be randomized. And the key on phase 1 dose finding trials is looking at finding what's the recommended phase 2 dose or doses that you should bring forward to the next phase. Now, you can imagine that as you move through the phase, the number of patients, the team resources increases as well as time and money. So it's really important to actually concentrate and do well in the early phase. And I also want to mention that there's increasingly similar phase 1, 2 as well. So it becomes, it's not so concrete about end of phase 1 moving on to phase 2. So sometimes it could be a lot of safe, similar phase 1, 2. Okay, so my focus is on early phase dose finding oncology trials here. So it's phase 1, so it could be first in human or first in combination of different therapies. And here the patients are advanced stage cancer patients. They are typically exhausted all standard treatment options. And so they're coming here and you really want to give them the best dose, which hopefully could be active. The primary objective is to assess safety and tolerability and to determine the recommended phase 2 dose. And that could be the maximum tolerated dose, and we heard quite a bit about it. So it's the highest dose that is tolerable, or it's the optimal biological dose that not only takes into account toxicity, but some sort of marker of activity. The key features are they're typically quite small, depending on the number of dose levels that you're looking at. So small sample sizes. In oncology, is typically non-randomized, but I think there are actually increasing randomized components within the dose finding oncology trials. I'll mention one later on. So Jack has given a great introduction about adaptive designs. So here is about adaptive dose escalation. So it's adapting the dose levels. You're starting off with small cohort patients, and you're talking about adapting after, like, say, 2 or 3 patients. And you want to find out what's the best dose to give to the next cohort patients. It's based on the observed effects, which endpoints are you using to make the adaptations. So the conventional assumption is, as you see from the horizontal axis, that's a dose level. So increase in dose level, you actually believe there's going to be an increase in the probability of toxicity, as well as an increase in activity. Now, this is obviously true for chemotherapy, so cytotoxic agents, and that's kind of the beliefs that's been the assumptions in the past. So the maximum tolerated dose is then the dose at which the dose-limiting toxicity rate is closest to your specified target level. So the DLTs is commonly defined as severe treatment-related side effects, and it's actually very much protocol specific. So different protocols have different definitions of what they would deem as dose-limiting toxicity. Say, for example, you decided to set the target toxicity level at 0.2, so as a trade-off to the potential benefit to patients, because you get as high as possible, then your maximum tolerated dose here is then dose level 3, and with a corresponding activity of, say, around 45%. But for example, you're combining two drugs, and one agent is giving already about 35% toxicity level, so you're giving two agents and hoping that you give much promising activity. Then your target toxicity level is 40% here, which gives a corresponding MTD of dose level 5, and a high activity rate about 70%. So key message is, very important to define what your dose-limiting toxicity is, and what is that target toxicity level, because it will determine what your maximum tolerated dose. And this needs a lot of discussion. And in fact, this is probably one of the things that needs most discussions with the clinicians, and also with looking at evidence to set the target right. So I need some audience participation right now. So because I know you just came from lunch, so if you're familiar with dose-finding trials, I would like you to stand, please. Ooh, let's see. Great, that's quite a lot. Great. Next, if you're familiar with rule-based designs, continue standing. Right, not much change. If you're familiar with advanced design, and we've heard some about the continuous assessment method earlier on, so whether that be model-assisted design or model-based designs, please continue standing as well. Great. As I'm speaking to quite a lot of experts here. Thank you very much, everyone. Great. I promised Andrew, who's the next speaker, that I'll wake everybody up. So hopefully that would. So let's look at what common designs have been implemented in practice. The most common trial designs is rule-based 3+3 because of its simplicity. As you can see from here, like just graphically, it's so much higher than other designs. But the implementation of advanced trial designs has actually increased quite a lot rapidly, from 5.4% in 2009 to 2014 to the latest review that we did, where we look at clinicaltrials.gov protocols obtained from there, and it's about 19% that has utilized it in the recent years. So there's an increase in the use, and I believe that you'll continue to increase as well. So the MRC Adaptive Design Working Group, we wrote this quick guide, why not to use A+B design. So it gives you a bit of details of why rule-based design is a lot more inferior to it. But the simple thing is, it is very simple. Perhaps you don't actually need a statistician, so in the past, it's been carried out without statisticians. However, there are a lot of disadvantages. And actually, in an early phase setting whereby every patient counts, you want to utilize all the information to make the best decisions. And that's why there's been a move to more advanced designs. Okay, I think a lot of you know about rule-based 3+3, but in case for some people who might not know, just a quick overview of what it does. And this is actually really simple. So the objective is to seek the maximum tolerated dose. You want to have 6 patients to be treated at the proposed MTD before you then declare that as the maximum tolerated dose. You typically start at the lowest dose, but you do not necessarily have to. And the maximum tolerated dose is defined as a dose with 0 DLTs or 1 or DLTs, where the next higher dose has 2 or more dose-limiting toxicity. So in this flow diagram, you can see that you give a new dose level to a new cohort of patients, and there's a traffic light system whereby green means it's safe, you're happy to escalate, so 0 out of 3, that's safe. Amber, 1 out of 3, you're unsure, you're going to stay at that dose level. And I believe that's how they got the design called 3+3. So you enroll 3 further patients at that same dose. And then at that dose level, you see 1 out of 6, that's safe, you're happy to escalate. But if you saw 2 or more DLTs out of 6, then you want to say that dose is unsafe, you not look at that dose again, and you have to de-escalate. At the beginning, if you saw at least 2 out of 3, you're saying that dose is unsafe, you'll de-escalate. And if that is your starting dose, your lowest dose, you actually stop the trial then. Now, what's the difference between that and the model and the model-based continuous assessment method? So the aim here is to seek a dose that causes severe side effects in a preset target percentage of patients. So say 20%, like what I showed earlier on in the graph. Now, how it works, you use a mathematical model, so it's called model-based, to describe how likely severe side effects are at each dose level. And after each group of patients is treated, all data is then used to update the model. And the model then helps to choose the dose that is closest to your target level of side effects for the next group of patients. And you can have practical constraint like without skipping of doses, but you could also allow that as well. So Emily, my PhD student, did quite a good illustration of how a CRM works, Bayesian CRM works. So you've heard from Jack about priors. So this is a case whereby we want a prior that is fairly flat across the different dose levels. So before you start, you're saying that any of the dose levels could be the MTD, could be the maximum tolerated dose. Maybe dose level 3 has just a slightly higher one. So you start off with that. That's your prior information. Now, and then you start the trial. You recruit 3 patients. So black means no DLT, and yellow means that there's a DLT. And the model just keeps updating. And you can see that the probability of the true MTD at any of the dose level changes over time. You can see later on that dose level 3 becomes more and more likely to be the true MTD, isn't it? So at the end of the trial, then that is likely going to be your maximum tolerated dose. You can see that the spread as well when you started off, is fairly flat, but now it's quite clear that dose level 3 has a high probability that that's the true MTD. So now move on to this first case study where we implemented the continuity assessment method in the Viola trial. So this was actually done probably about, I think, almost 15 years ago now, actually, when we first sat down to discuss this trial design. And one of the reasons for actually moving from a traditional 3+3 to a continuity assessment method is that we have a smaller sample size. We have 7 dose levels. How do we maximize and really utilize all the information as possible and get a high accuracy in identifying the right maximum tolerated dose? So in this case, we are trying to look at combination of lenalidomide and azacitidine for patients who relapsed after allogeneic stem cell transplantation for acute myeloid leukemia. So it's looking at combination therapy, targeting an estimated toxicity rate within this first cycle of treatment that's closest to 20%. And we use a one-stage Bayesian CRM. Now, I have to say that when we first did this, it was the first time there's no information. It was the first time people have done it in an academic trial setting. So it was a bit of a leap of faith, even though in the background, we've done loads of simulations to look at various scenarios to make sure that the design works well. But it's still a leap of faith, and I'm very thankful to Charlie, who actually gave me the confidence to actually run this trial with me. Okay, this is actually what happened in the actual trial. So this shows you the first 3 cohorts of patients. And you can see that the prior curve here, that's the prior curve of the prior information estimated DLT rate at each dose level before we started. So before we actually see any data. As you recruit patients, so for example, cohort 1, we started at dose level 0. So there's 7 dose levels. There was no dose limiting toxicity. If there's a DLT, it's in red. We then escalate to dose level 1. And the prior curves keeps updating with all the information. And in this trial, we did not allow skipping of dose level. So even though we could have skipped and gone down higher, as shown from the curve after cohort 1, but we did not. We stayed because we did not want to skip doses. So cohort 2 were dosed at dose level 1 and did not have any DLT. Now, this is an interesting incident that happened. Cohort 3, all 3 patients were supposed to get dose level 2, but 1 patient was actually incorrectly dosed at dose level 3. And so that happens in real life, that actually things do not go according to plan. And a lot of things do not go according to plan. You can plan as much as you want at the beginning for a clinical trial, but actually a lot of things there will always be surprises. So in this case, in a 3+3 design, it's going to be very tricky because you only can make decisions after 3 patients. But in the continuity assessment methods, the model-based design, it doesn't matter. You're just taking all the information to date and update your model. So that's what we did. And in fact, the rest of the patients actually got dose level 3. There were 2 DLTs at that dose level, and the dose level 3 was declared as MTD because that's a dose with the estimated DLT rate that is closest to our target of 20%. So we did the job. So what's the highlights for this? I think we saw effective handling of incorrect dosing, and also the dropouts that I did mention, whereby this group of patients were actually very sick. So some of them never actually finished the DLT period, so we had to keep replacing it. So we actually have a lot more flexible enrollment in the later part of the trial. So that accelerated that recruitment and saved time and resources. And the good thing was majority of patients were enrolled at the recommended dose, which is dose level 3. And as I mentioned, before we actually implemented, we had to do loads of simulations, and compared against the 3+3, there was definitely higher accuracy in identifying the right dose. So while trying to implement the CRM, I think one of the key things we're aware that it was published in 1990. So it's been there out in the literature, but it was really difficult for people to implement it in practice. And that's particularly because it seemed like a statistical black box compared to a 3+3 design, which is so much simpler. Like you see the flow diagram that I showed earlier on. And so that really motivated this particular practical tool called the dose transition pathways. And the key thing is that it tries to predict, or in fact, it just predicts future doses for upcoming patients using all the accumulated information. In this case, either you stay at the dose, you escalate to a higher dose, you de-escalate to a lower dose, or you stop early. So this is what the initial dose transition pathways for the first 3 cohorts look like for Viola. And in fact, you can map this out right at the beginning of the design stage. And it's actually very useful to do that. Why? Because you can show that it's a similar flow diagram that 3+3. You can show that to your non-statisticians who might understand all the different parameters that you need to calibrate in your model-based designs. But what is useful is we're going to look at the flow diagram and how the dose will be recommended for the design. So say, for example, we're going to start at dose level 0. There's no DLT, 0 out of 3. Cohort 2, you then give dose level 1, and then you escalate again if there's no DLT to dose level 2. However, if there's 2 out of 3 DLTs, 2 or 3 DLTs, you then de-escalate to dose -2. That's the lowest dose. And also, it's the next discussion is when you're at the lowest dose, which is dose -2, how many DLTs out of 3 patients? But if you see that, you'll definitely stop. And that's really a discussion that is very helpful and ask the clinicians if in practice, if they saw that number of DLTs out of 3, so whether it's 1, 2, or 3 DLTs, they will actually stop. So the answer was 2 or 3. And we then calibrated the design, making sure, so there's an underlying Bayesian stopping criteria there. So we calibrated it such that it will actually be that you will stop when you saw 2 or 3 DLTs out of 3 patients. So it can be a design tool, but it also can be operational too, because it actually just pre-analyzes the different outcomes for you. So you can put that into your SAP or even the protocol. And that's how it runs if you recruit in cohorts of 3. And obviously, if the cohorts change in size, you can keep updating those transition pathways and see how the design will recommend for the subsequent group of patients. So that really helped. And in fact, when I first showed this to the trial manager who leads that operational team, then she was really relieved. And that really helped us to realize that this was the right direction, that we need to make things a lot simpler. It also gives us the confidence that whatever we calibrate is something that is fit for purpose. So it's transparent, it aids understanding, and we can include that in the protocol. So for any proposed dose, a complex dose finding design that you can come up with, I think it's very important that we obviously have to look at the design performance. So you look at various clinical scenarios, and in this case, you want to make sure that there's high accuracy in finding the right dose, in finding the true dose. And then also look at the initial dose transition pathways, particularly for complex dose finding designs. That's very helpful because you're actually going to implement it in one real trial with real patients. So you really want to make sure that it is fit for purpose. So now I'm going to move on to a case whereby what if the treatment activity may not increase with dose? And we're increasingly seeing that for target therapies or immunotherapies, whereby as you increase in dose level, they might all be very safe. So the maximum tolerated dose here in dose level 6, but actually the activity might plateaus. So you can have the red curve or you can have the green curve, and the optimal dose might not be dose level 6. So you need to consider other outcomes to help you make that decisions. So for example, you could be using treatment response. Now, we were faced with this problem where we're trying to design the match point trial. And because it was, they were looking at 4 dose levels, we asked clinicians whether as you increase in dose level, are we confident that actually activity will definitely increase? And they told us, in fact, of a few states, all agreed that actually those level 3 and 4, they're not sure that they think that there might be a plateau effect there. So we then think about, okay, we need to not just look at, so CRM won't work, the 3+3 won't work. We think about a design that actually has an efficacy and toxicity trade-off to inform what's the best dose to bring forward to the next phase. So, the trial design paper that was led by Christian Brock. In fact, he enjoyed doing it so much that he did a PhD, and that was one of his PhD chapters. And the trial results paper was just published not too long ago. And Murray Copeland was the chief investigator. In fact, this is again a leap of faith. It was a complex design, a lot much more complex than the CRM. In fact, it was actually designed probably a few months just after the Viola trial. So this is the results of the actual trials. There's really small number of patients, as I mentioned. Patient number is really small. We defined the optimal dose that balances activity and toxicity. We had 4 dose levels. We started at dose level 3, which is believed to could be the highest dose that is tolerable. Now, you can see for the first 3 patients, if you just concentrate on that, we did see 2 DLT out of 3 patients. But the 3rd patient who had the dose-limiting toxicity also had activity. Now, if we look at just using a 3+3 design, we'll just actually stop the trial at that point. Or not stop the trial because we started at dose level 3, we'll actually drop the dose to dose level 2. But however, using the F-TOX, all the patients actually receive the same dose throughout the trial, balancing both activity and toxicity. So it makes a difference when you actually determine which are the endpoints that you're going to use to make the interim decisions. So we actually saw a stronger response rate than expected, about 68%. We avoided lower recommended dose. As I mentioned, we've gone down to dose level 2 if you use a conventional 3+3 design. And also this trial was actually really hard to recruit, and I did not anticipate how rare the patient population is. And we started off with cohorts of 3, but later on we make it a lot more flexible, and we could just allow continuous recruitment because every patient counts and we want them to come in as soon as they could. And as I said, before you do any designs, you have to do lots of simulations, lots of virtual trials to check that the design works well in practice, works well across various clinical relevant scenarios. And that's what we did. So in fact, this is the first prospective trial of plastic transformation in chronic myeloid leukemia, and the results were actually incorporated into the European guidelines. So even early phase designs can make a difference. So next, my last case study was a phase 1 CAR T trial in a relapsed B-cell lymphoma here. So this is actually still being set up. It's funded by CRUK, and I have 3 lovely clinical investigators who work very closely with me on this. So the objective was to evaluate the safety, efficacy of the next generation anti-CD19 parallel CAR T. So the challenges here is that, as some of you might know, the CAR T cells are very costly. We're trying to look at up to 5 doses. We only have 18 maximum that could fund 18 available patients within the funding period and the funding pocket. Higher doses may not be more active, and that's been seen in the previous literature, and the activity may be delayed. So we then think about, okay, we can't do what we did earlier on, like the last 2 designs. We need to come up with something different. So we proposed a randomized Bayesian dose optimization design to find the best dose for balancing side effects and treatment benefit. And Cynthia, the trial statistician, is actually here if you want to speak to her more about designs, and please feel free to. Next, I'm going to move on to my next topic about incorporating patient-reported outcomes. So I mentioned very briefly, patient-reported outcomes are actually rarely implemented in early phase setting. What are patient-reported outcomes? So PROs are defined as any report of the status of a patient's health condition that comes directly from the patient without interpretation of the patient's response by a clinician or anyone else. So we looked at, in dose-finding trials, the dose decisions are typically done by tolerability and activity. We saw that earlier on with the examples. And often PK and PD can come into it and make the decisions. But the question is, can patient-reported outcomes play a role in this setting too? So PROs are the gold standard for assessing symptoms, physical function, and quality of life. While common in later phase trials, their use in early phase trials remain limited. So in fact, in a review that we did, where I look at more than 10,000 trials and clinicaltrials.gov, even though this is probably 2007, 2020, probably quite long ago now, it's 5.3%. I believe it's increased over time, and we actually saw a significant improvement or increase over time, even during that period of using patient-reported outcomes. But most of them are used as secondary endpoints. However, there's been growing interest from patients, academics, industry, and regulators in the use of patient-reported outcomes in the early phase settings. You can see from all these papers here that there's just a few selection of those, and that's just been published in the very recent years. So, and really thinking about how do we use PROs in dose optimization trials? How do we utilize, what PRO should we be collecting it? How often? So in fact, in this very recent papers that we have, so we invited like a group expert virtual roundtable of patients, clinicians, regulators, both in oncology and non-oncology, to actually really discuss how do we best utilize PROs in that setting. And it's one of the, I mean, so we came up with some recommendations, but I think one of the things that I brought that it was quite clear was PROs are rarely used in oncology, even though it's probably a bit more advanced than on non-oncology, but lots of things that could be done in this space. So that actually, and that's partly, there's a very current program called the Optimize program, which is incorporating patient-reported outcomes in dose finding trials. We've been looking at what are the research objectives that we should be having if we utilize PROs in this setting. What are the designs that we could be using? And should we utilize the PROs to actually help interim decisions, or do we utilize it right at the end of the trial to find the recommended phase 2 dose? And once we know, and then I guess right in the center is the pro-selection, which is one of the, when we did a survey with stakeholders of what are the key challenges and key barriers, I think this is about the pro-selection, because what should we be collecting? Particularly in the early phase, a phase 1 setting, whereby you might not know the symptoms very well. How many symptoms, pro-symptomatic adverse events do you want to collect? And one of the good things about patient-reported outcomes is that because if you're asking those questions, patients are thinking about it, and sure they will answer that. And that's been shown to collect more information compared to what the clinicians report. So actually collecting the number of items and what items you collect is actually quite a critical bit to solve this puzzle. So the optimized P is actually, it's almost like a study within a trial for people who knows about SWOT. So it's a pro-platform study to integrate patient-reported outcomes across multiple dose-finding oncology trials. It's going to open soon. And so if you're interested, please come and speak to me. You can see from here, there's a space here. In fact, there's loads of space. So please get in touch if you're interested in this area about incorporating patient-reported outcomes in early phase trials. So the key takeaway is that different designs can result in distinct interim and final dose decisions, underscoring the critical impact of design choice. So really important to spend time thinking about and really speaking to various experts to design the trial to make sure that you actually use the right design. And this paper, it looks at would the recommended dose have been different using novel dose-finding designs? And the answer is yes. You've already guessed. So it really requires teamwork, and it's not something like you can do it within a few hours. You really need to think carefully about what's the best design for your trial. The next part 5, I'm moving on to good reporting in early phase trials again. So, and this is talking about good reporting in trial protocols and trial results publications. As the late Professor Doug Coleman, who pioneered the consult work, so readers should not have to infer what was probably done. They should be told explicitly. So let's look at the reporting quality of dose-finding trials. So we did a random selection of 106 protocols from clinicaltrials.gov in 2017-2023. The percentage of components that adequately reported, so it's just some of them. The escalation and de-escalation criteria, 74%. Definition of the safety analysis population, 66% reported that. And in terms of the dose escalation analysis population, so which patients, like what's the definition of the patient that you'll be using to help to make those decisions, that were actually only defined in 34% of the trials. So it's inadequate reporting in trial protocols. And when we look at the results, so this is trials publications, and we did a random selection of 476 published trial reports between 2011 to 2020. Again, you can see here very poor and inadequate reporting. So 29% talk about what they plan to maximum sample size. The rationale for starting dose, which is actually a very important component, were actually reported quite poorly in this setting as well. And most of such trial results did not have an accessible trial protocol. So even though you want to know it, you wouldn't be able to find out. So inadequate reporting in published trial reports. So there's time for a change. We really need international consensus-driven guidance to provide clarity, reproducibility, informativeness, and the utility of results. So this is to recommend essential items for protocols, the extension of the spirit, which is for randomized trials, to the dose-finding settings. So it's the spirit defined, as well as the trial reports for consort defined. So what we did was we did a Delphi survey followed by a consensus meeting, and we had over 200 participants from over 24 countries to help shape the consensus guidance. So very much thankful for this because each survey actually took really long. There were lots of unique features about those funding trials compared to the randomized settings, later phase trials. So here the pie chart shows you the different stakeholder groups that were participated in it. So majority were clinicians and statisticians. But we had regulators, patient partners who participate in this as well. So the good news is that the hard work has been done. So these are the new reporting guidance for early phase dose-finding trials. So we've got the checklist, the protocol guidance, and the trial for trial report as well. And so we've got practical examples. So these are exemplars of either from protocols or for trial results that reported those items well. So that will help people to be able to utilize that to actually create your protocols as well as create your trial results publication. So very thankful for all the people who actually participated. You can see the list of authors are huge. So the key highlights of these two new checklists is that the new items are, for example, the key findings from relevant non-clinical and preclinical results. As you can see, those finding trials are very useful, important things. The range of dosing regimens, including starting dose levels with the rationale behind that. The planned cohort size, are you doing a fixed, a flexible, adaptive? Because that will have different performance as well in terms of the design. And then interim results used to inform decision-making. For example, what you used to inform your dose escalation or de-escalation. Now, these are new items. The modified items for the original Spirit and Consort is about pre-planned interim decision-making criteria and stopping criteria for dosing and other trial adaptations, the statistical methods in terms of adaptive features, or your analysis population that are being used for your interim and final. And reporting key results by dose level. That shouldn't be a surprise to everybody because this is dose level trials. We want to report your key outcomes by dose level. So the two brown ones are specific to reporting of trial results. So that's under consort defined. So this just gives a snapshot of what the consort checklist looks like. So we've got standard consort statement here. And then we've got the consort defined for dose finding trials. Now, if I look at a lot, because it's one modified items, with the 10 new items, but actually it's just very unique features. For example, starting dose with a rationale, what is your range of planned dose levels with rationale. So it's a sub-items under the trial design category because we need more information about the unique features of those findings. And just to mention that is also applicable for both oncologies or disease types span through. So now I've got a question for you. What level, so you've seen the level of reporting quality and earlier on in protocols as well as trial results. So what level of improvement in reporting quality would you like to see in 5 years' time? So you have to choose one. And I've not put none because I believe that everybody's positive that there should be some form of improvement. But there's 4 categories here. So at least 10%, at least 20% that you want to see, at least 30%, or you reach perfection. So shall we start with at least 10%? Anyone? Okay, at least 20%. Great. Okay, 2. At least 30%. Okay, great. Reach perfection. Wow. That's a great aim to aim. Okay, so you have actually committed to that. So hopefully in 5 years' time, all of us play a role to actually make a difference. I think whether you be writing your protocols, you're writing trial results publication, or you're doing peer reviewing, or you're an editor, I think it's really important to encourage people to report their trials well, or actually to do high quality trial protocols. Okay, so I'm not going to touch very much on it, but Andrew's going to talk a bit more about actually this lay summary work that we also did as part of this DEFINE project, thinking about how to explain the results in a very friendly way to patient partners. And that's really very much motivated by our patient partners. So this is actually a presentation that Andrew did at ICWMC. Okay, 6 key takeaways. So impact. So designing trials that truly matter. Influence I. Trial designs that shapes both interim and final dose selection designs. I hope convince you that actually it really makes a difference of how you use the endpoints and what design you used. Map. And when you design early phase trials, you not only look at that trial that you're designing, but actually really see about how does that going to influence, what's the next stage, how they can influence your later phase, and make sure that you actually utilize all the outcomes, collect those outcomes that can help inform the decisions. Pros add values, so pros can complement traditional safety and efficacy measures, offering a fuller picture of treatment effect, whether that be tolerability or treatment benefit. Aligned, ensure the design supports the trial goals to enhance design accuracy, and we've heard that earlier on in the earlier talks as well. Collaborate, I think Pavel mentioned, that's so critical. So foster strategic partnerships across different disciplines. There's so much that we can learn from each other to strengthen expertise and sustainability. And trust about good reporting. Transparency and clarity in conduct and reporting is essential for credible research. So this is my incredible team. So a lot of things I talk about, especially the current work, are really under the operational side. They're all running the show. So they are the people that I'm really very much grateful for. So we've got PhD students, we've got trial methodologists and statisticians as well, and working on it, and a few other familiar faces as well. So there are exciting opportunities that, hey, if you want to think about career change or something like that, so we'll be soon recruiting for statistician, methodologist, senior, all across different levels as well, including group leaders. Or if you're interested in internship or placement to find out about what we do, please get in touch. So thank you very much. So to end off with Doug Ottman's quote, to maximize the benefit to society, you need to not just do research, but do it well. And I talk a lot about team size, and it's so critical. And I just want to acknowledge the different people that had the privilege to work with, and these are all listed here. Yeah, because I've learned so much from everyone. Thank you.